This is a new series that we've started highlighting key scientific advances in the CMT space. We do have a few announcements I wanted to share before we get going regarding our convention in September. So if you enjoy the conversation today, we'd love to see you at the 2026 Global CMT Research Convention, September 24th to 26th in Cambridge. It features 2 days of a research-focused program with a third day of patient-focused programming. So you'll get a chance to interact with both leading researchers and patients looking to our research for hope. For those attending the convention, we have recently extended the abstract deadline for oral presentation, so we're now accepting abstracts through August 10th. It's a great way to present your research in front of other scientists, industry leaders, and venture capital representatives if you're interested. Another specific opportunity, if you're within 10 years of your terminal degree, whether that's an MD or a PhD, you can also be eligible to get free registration by applying to the Emerging Researcher Forum, which is held the first day of the convention in the morning before the official programming kicks off. We have a limited number of travel stipends, so if that's something that you're interested in, please apply on our convention site. And then if you're not already registered or if you have questions, we can help with that too if you want to reach out. And we're planning on hosting a webinar at 7 p.m. Eastern on August 6th to explain some of the benefits of this year's convention, as well as to answer any questions that you have. So with that, I'll kick it over to Dr. Hershey Condetti, who will be introducing our speaker today.
All righty, good morning, everyone. Thank you all for being here for the inaugural CMTRF Journal Club series session. And so today we'll be listening to Dr. Marina Stavrou discussing some of her latest work. And so Marina is an associate scientist at the neuroscience department of the Cyprus Institute of Neurology and Genetics in the lab of Professor Kleopas Kleopa. And so her primary research focuses on developing novel translatable gene therapy approaches to treat CMT1A neuropathy. And we are extremely excited today to listen to her share some of her latest work on AAV9 RNAi therapy for CMT1A. And so with that, please take it away. We're really excited to hear about it.
Hello, thank you all for joining. And let me start with sharing my screen. So again, hello everyone. Thank you for the invitation to present our recently published work on AAV9-mediated RNA therapy for CMT1A. The title of the paper is Safety, Efficacy, and Distal Nerve Schwann Cell Biodistribution in Mice and NHPs to support the translation of AAV9 RNAi therapy for CMT1A. This paper was published in Molecular Therapy Nucleic Acids. So I will just briefly touch what CMT1A, as I guess we are all very familiar with the term. So CMT1A is the commonest type of CMTs and results after the duplication of PMP22 gene. Overexpressed PMP22 creates a gene dosage effect that destabilizes the myelin sheath, leading to demyelination and secondary axonal degeneration in distal and proximal nerves, as well as in the spinal roots. Like all CMTs, currently there is no approved treatment for CMT1A, but as we're all aware, there are many emerging therapies, gene and drug therapies that are very promising, and I am very optimistic that we're going to witness the event of the first treatment being accepted and going out to the market. So in collaboration with Scott Harper's lab, at Nationwide Children's Hospital, we previously published a novel gene therapy method that acts at the RNA level. I'm not sure if you can see my mouse. And prevents the excess PMP22 protein overexpression. In detail, we developed an artificial microRNA, which is MIR871, that we packaged into an AAV9 vector, and we intrathecally injected it into a mouse model of CMT1A. Mir-treated animals showed therapeutic benefit at early and late disease stages without causing any safety concerns. And this proof of concept project was funded by CMTRF. The program is now licensed by Armadus Bio and has already reached key regulatory milestones, including the FDA Orphan Drug Designation and the Rare Pediatric Disease Designation. So, building on this previous proof of concept study, the newly published paper focuses on the key questions needed for translation. The first question was safety at the sequence level. Does MIR871 specifically silence PMP22, or could it just affect other human transcripts? To address this, we performed an in vitro on and off target assessment in 3 relevant human cell types: neurons, hepatocytes, and Schwann cells. These are important because they represent tissues that are mostly exposed after the intrathecal AAV9 delivery. The main finding was that MIR871 has strong on-target silencing effect on PMP22 with no evidence of significant sequence-specific off-target activity. We then developed our second generation vector that is more translatable, as it exclusively expresses MIR without the CMVG reporter gene. The CMVGFP reporter was used in the JCI paper, and now for this new molecular therapy paper, we excluded that. Our previously published work was also performed using only one dose of the AAV9 MIR. Now for this paper, our second generation vector was used at the dose escalation, and we tested the safety and therapeutic effects of different doses at 6 weeks and 4 months post-injection. At 6 weeks post-injection, we first assessed whether different vector doses reach relevant tissues in mice. So now we're looking in mice 6 weeks post-injection. Data show that there was a dose-dependent transduction of neural and peripheral organs, with only exception being the liver, that showed lower vector genome copy number scores with the highest dose. At the same time point, we also confirmed the expression of our mature MIR sequence at neural and other tissues. The most consistent dose was 5E11 VG per animal. This is the same dose as the one that we used at the JCI paper. Even though the highest dose had highest vector genome copy numbers in some tissues, this did not translate in better MIR expression in the PNS tissues. We then proceeded with testing the silencing efficacy of each dose at the mRNA and the protein level, again in mice, and concluded that the most consistent dose is 5E11, as pointed here with the yellow arrows. We also looked at the MPZ, myelin protein 0, which is a major myelin protein that is an indirect indicator of the myelin sheath at the molecular level. At 5E11, MPZ levels increased, supporting an improvement of the Schwann cell and myelin status. So at this stage, 5E11 stood out as the dose with the best molecular profile, effective PMP22 silencing, together with signs of improved myelin profile. One important concern with PMP22 silencing is that we don't want to over-silence. PMP22 knockout can cause HNPP. So the goal is not the complete knockdown, but the normalization of the PMP22 levels. To address this, we performed a dual, custom-made microRNA RNA scope analysis, allowing us to detect MIR871 and PMP22 transcripts simultaneously at a single cell level analysis. Dual microRNA RNA scope analysis is basically an in situ hybridization assay for the detection of our microRNA and PMP22 RNA in fixed mouse tissues. We applied this technique in PNS tissues of animals injected with the two of the highest dose of AAV9 MIR, 5E11 VG per animal and 1E12 VG per animal. Looking at the hybridization pictures, we conclude uniform expression of MIR871 at both doses. If we go down to the bottom graph, we see that this uniform expression is followed with a dose-dependent decrease of the number of PMP22 dots per cell, i.e., a dose-dependent reduction of PMP22 RNA levels. Importantly, though, if we go to the top graph, we see that the number of PMP22 positive cells remains unchanged if we compare the PBS treated animals with the AAV9 MIR treated animals at both doses. This is a positive result because it confirms that even though we silence the amount of PMP22, the amount of dots per cell, we don't totally delete PMP22. This supports a controlled normalization and reduces the concern of an HHPP-like over-silencing. We then move to functional outcomes. So mice were treated and analyzed 4 months post-injection. 511 was also the dose with the strongest therapeutic benefit as it resulted in the best functional and electrophysiological performances. This is important because these are translationally relevant readouts. They are not only mouse behavioral outcomes, but they are closely related to the measurements used in patients. We also analyzed blood safety parameters and serum neurofilament light levels. In blood analysis, 5E11 was the dose that resulted in the greater decrease of the NFL axonal degeneration blood biomarker. Also, a full hematology and serum analysis showed no difference among groups. And here we show the indicative liver enzyme levels that confirm the safety of our approach in mice. Together, these data support that 511 is not only effective, but is also very well tolerated in the mouse model of CMT1A. We then assessed PNS tissue morphology. Here we show lumbar motor roots as the most indicative tissue. CMT1A mice at baseline before treatment, they have a very characteristic nerve pathology, including demyelinated fibers, thinly myelinated fibers, and onion bulb formations. 5E11 was the dose that resulted with the strongest morphological improvement in neural tissues, and here we see the lumbar motor roots as the most prominent example of all the tissues we analyzed. So we analyzed the lumbar motor roots, the sciatic nerve, and the femoral nerve. The highest dose now, the 1E12, did not provide any additional benefit when compared to the 5E11. In fact, across several readouts, 5E12 was less effective than 5E11. So, all this time, we have been doing experiments in mice when our ultimate target is humans. I think this image can convince you that mice can be a perfect model, but still highly different from humans. Until now, we have been intrathecally injecting mice, and the most distal part to reach was 2 centimeters away from the injection point. In humans, the intrathecally injected product should travel about 1 meter away from the injection point to reach the distal nerves. This distant problem is critical for CMT1A because the disease affects long distal peripheral nerves. As you can expect, we cannot just jump from mice experiments to human testing. That's why we continue with performing a very detailed dose escalation study in non-human primates. Based on our murine dose escalation study, we extrapolated the 2 most effective doses, 2E11 and 5E11, to proceed with the NHP testing. Extrapolation was done based on the CSF volume of the animals in order to demonstrate the scale-up potential. So we intrathecally injected NHPs with these 2 doses of our AAV9 MIR vector and analyzed animals at either 6 or 12 weeks post-injection. We performed the standard weekly and neurological observations and collected weekly samples for blood and urine analysis. At the final time points, we performed all the tests that you can see listed here, including molecular like DDIT, PCR, Western blot, PNRS morphology, and many more. So the first major question in NHPs was, does the vector reach the tissue of interest? So what happened in mice can also happen in NHPs. And the answer was yes. Here we see the vector genome copy numbers at 6 and 12 weeks post-injection. We see a summary of a very detailed vector genome copy analysis that started from the brain and finished down to the sciatic nerve. Here we see all the neural tissues in this image. Focusing in the 2 line graphs at the bottom, we see that the closer to the injection point, that is pointed here with the yellow arrow, the higher the vector genome copy scores, and that the vector genome copy scores diffuse from 6 to 12 weeks post-injection without clearing out. We did not observe a clear low versus high dose response in terms of the vector biodistribution, but the key translational point here is that the vector reached the distal PNS tissues after the intrathecal injection. This is something that was shown for the first time. So these numbers reflect the amount of vector, how many AAV9 particles are in each nerve. But what happens in the actual amount of the microRNA, which is our actual payload? Importantly, regardless of this viral diffusion, the average miR-871 expression remained relatively stable between 6 and 12 weeks post-injection. We detected our mature MIR sequence in all peripheral nerves, and this suggests that functional expression can persist even when the vector and genome measurements decrease over time. The next question was, okay, we have the AAV9 vector, the vector is there, microRNA expresses, but does miR-871 silence PMP22 in non-human primate nerves? We observed PMP22 RNA and protein reduction in all the NHP peripheral nerves that we analyzed at 6 and 12 weeks post-injection. Here we see the sciatic as the most prominent example. The MPZ remained unchanged, and this is very important because it supports that target engagement exists, but does not come with the disturbance of this major myelin protein in NHPs. So remember that our primate, it's a normally expressing animal. It has normal PMP22 levels. Then the other question was safety. The NHP study included a broad clinical, pathological, and safety assessments, including ophthalmic examination, ECGs, neurobehavioral observations, anatomic pathology, clinical pathology, blood measurements. All safety analysis included to no article-related toxicity. And here we show the indicative liver enzymes at 6 and 12 weeks post-injection. This is a key translational finding because it supports the tolerability of the lumbar intrathecal AAV9 MIR in a large animal model. We also specifically focus on DRGs and peripheral nerves from the potential toxicity. So DRGs are very important because if you're into the topic, you know that there have been previous literature suggesting DRG pathologies after the AAV9 injection. So here we see the representative H&E images from the cell line controls and vector-injected animals at both doses and both time points that confirm no article-related pathological changes. Here we see 12 weeks as the most prominent example, but exactly the same images exist for the 6 weeks post-injection. Semitting analysis on HNP PNS tissues showed no evidence of treatment-related nerve pathology and definitely no evidence of an HNPP-like phenotype. This supports, again, that we do have PMP22 silencing, but is not accompanied with the excessive knockout or HNPP pathologies in a normally expressing animal like a primate. Finally, we assessed nerve electrophysiology in NHPs. Again, there were no significant changes in nerve conduction velocities or CMAPs after the treatment, and this supports the functional safety of the treatment and it is consistent with the histological findings that we showed in the previous slides. So across clinical observations, blood and urine analysis, pathology, histology, electrophysiology, all NHP data support a very strong safety profile. So coming to the conclusion, in this paper we showed for the very first time a very detailed PNS biodistribution analysis of an intrathecally delivered AAV9 vector. This IT-delivered AAV9 vector, we show for the first time again that it does reach the distal sciatic nerves, and by showing the PMP22 silencing, we indirectly show that it also reaches the Schwann cell specifically. Again, we show for the first time that we silence PMP22 without causing the HNPP-like phenotype. This is something that happened for the first time in NHPs, but we have also showed that in our JCI paper, a very similar phenotype that when we injected normal wild-type mice, we showed the silencing of PMP22, but no electrophysiology or any myelination abnormalities. Both mice and NHPs analysis suggest a widespread AAV9 biodistribution, a stable miR871 expression, no article-related toxicity, target engagement across the species analyzed, and no HNPP-like phenotype. Altogether, we're very confident to say that these preclinical data support that intrathecally delivered AAV9 miR871 is a scalable, translatable, efficient, and safe gene silencing approach to treat CMT1A. Coming to the end, I wish to thank my supervisor, Professor Cleopa, and our collaborator, Professor Harper and Lindsay, who have equally contributed in this project. We wish to acknowledge our valuable sponsor, Armadus Bio, who licensed also the patent of this project, and everyone for being here and hearing our presentation. Happy to take any questions.
Okay, if there are no questions in the chat, I can go ahead and start.
There's one in the
Um,
Q&A, actually. It's
oh,
a
is there?
different function. Yeah. So I
Oh,
can read it if you can't see it.
yes, please. It is not popping up for me for some
Yeah.
reason.
So someone asked, I'm curious about the variability in nerve conduction in the control samples. They appear to drop and then improve between 6 and 12 weeks. Do you know why?
No, again, this comes to the no article-related toxicity. So we don't know why. They're NHPs. It could be a technical issue during the examination, but no.
Mary, you have a question?
Yeah, thank you so much for a very nice overview and a very nice study. My question relates to, any update on where the kind of therapeutic development of this modality is. And, second question, the microRNA binding site is sequence-based. So do you know if there can be any kind of sequence heterogeneity in the patients that might affect the, I guess, potential of the therapeutic?
So starting from your last question, our miR871 targets a 3' UTR sequence. So there's a paper from the Mary Riley group from UCL telling that there are CMT1A patients that have a specific mutation in that region. So these patients might not have the same benefit as the other patients. But again, our microRNA also binds with mismatches. So even if it's not on 100% complementarily, it can still bind and silence. We need to test that. But we know that when, let's say that there will not be this group of patients, they will not be the first people to test and actually see in a clinical trial if the therapeutic effect of this approach. Ideally, we start with the patients that they do not have this mutation in their 3' UTR. At the moment, as I said in the presentation, the patent of this product is licensed by Armadus Bio. So they're handling everything that has to do with the translatability and collection of funding and proceeding to the clinical trials. It's a long process. I mean, the first paper our work started back in 2018, but back then there was absolute no hope and no other treatment. So 2026, we're here and we know that there are also many other groups that they're doing great work. And I think our patent is in good hands and we hope to collect the funding needed to proceed.
Thank you very much.
Thank you.
Great. We have a question from Brendan, so I think this should allow you to talk.
Hi, Marina. This is Brendan's daughter. Really great, great presentation. I'm obviously very interested in this story. I had a quick question. I was wondering what you think about the kind of non-length dependence of your biodistribution, and if you, maybe I missed this in the primate data, but how much of the virus and the gene product are ending up in the liver and other peripheral tissues, and do you think that maybe some of the distal transduction you see is virus that's escaped, into systemic circulation or lymphatic distribution and then entered the nerve peripherally as opposed to kind of, moving through the nerve itself?
Yes, so starting from our previous paper and other papers of the lab that compared the IV and the IT injections in mice, we know that this is dual. So when we intrathecally inject, the virus reaches the distal sciatic nerve through osmosis and the different pressure between the spinal canal and the nerves, but definitely there's a virus that escapes and gets into the circulation through the microvilli that exist in the area. So we know that it's dual. And yes, we had viral vector genome copy numbers in the liver and in other tissues as well.
Thank you.
Thank
If
you.
if there's time, I have one other
Yes.
question. In the NHP, did you look in I saw the histology that you showed of the DRG and the nerve. Were you able to see either with, RNA scope or something like that, histological evidence of your construct expression? And if so, did you look at different points in the nerve and how kind of robust the expression was at a histological level?
No, we didn't did the RNA scope with NHPs for clear financial reasons. And but we concluded this Schwann cell as that the microRNA enters as Schwann cells through the silencing of PMP22. So there was no chance to silence PMP22 if the microRNA was not in the Schwann cell. That was an indirect way to confirm the Schwann cell penetration.
That makes sense. Thank you.
Thank you.
Okay, we have another question in the Q&A chat. How long do you expect the AAV episome to last in the Schwann cells?
That's the $1 million question. So we know that in mice, it can last for up to 1 year. We need to follow this research more. So in our NHPs, we had vector genome copy numbers until 12 weeks post-injection. But we saw numbers, so I don't know for how long they would stay there. But we do have the SMA story, and we have itivizumab now. And so this is basically the SMA gene therapy product that has it just been accepted by the FDA to be delivered intrathecally into kids. So this is a very good example and a reference point for us, even though SMA sounds like a not very CMT relevant disease to us, it's an absolute reference point.
My laptop is frozen. I am curious about the dosing that you saw in the NHP. So you mentioned that there wasn't a significant difference in the biodistribution and the knockdown. I'm curious how, when thinking about moving that forward into human studies, how you might approach dosing that way when you're not seeing this difference, especially when considering potentially administering too much or too little to have that effect on the PNP22.
So at the moment we're considering with moving these two doses extrapolated, but it seems like there might be a space that we still need to investigate a bit higher, a bit lower. And we definitely again refer back to the SMA division story. So we see how their intrathecal injection will go, and we decide. But if we are concerned about the oversilencing effect, this is the most serious concern that someone may have for treating a CMT1A patient with a gene therapy product. We have seen that normally expressing animals, so animals that have normal levels of PNP22, mice and NHPs, have, after a treatment, show silenced PNP22 levels, RNA and protein level, but they do not show electrophysiology problems or myelination problems. This makes us believe that Schwann cells are very smart cells, let's say. So they can auto-regulate and they can compromise with over-silencing, and that's one. And the other thing is that HNPP might be a developmental disease. So there's no evidence supporting that an adult with a fully developed PNS system can get HNPP. So if dosing concerns are related to the NHP side effect, we're pretty confident that this might not be the case. This is not the most likely scenario. If dosing then has to do with the need, the dosing concern has to do with the need of repeated dosing, if I need a boost, or if this is not enough and then we need more silencing, there are projects that they now investigate the second dosing effect with non-viral platforms, like redosing with non-viral articles. But again, this is something very new, and many people have tried many different non-AAV9 vectors, many different AVs, but at the end of the day, AAV9 is always the best. We are here and we will see what the future will get us.
Another quick question. So I know you talked about measuring PNP22 expression, looking at the nerve, but do you think that in the future, as you move towards clinical trials, looking at skin biopsies would be useful? And has there been any success there in measuring PNP22 levels at that level?
Yes, there is a paper from John's Varen groups, and they do measure the PNP22 levels using a technique called nanostring. So this is an indirect way to measure the PNP22 levels in humans. There are also many projects that they're trying to indirectly measure the severity of the disease using blood biomarkers. So either trying to find myomeres, microRNAs, or different markers like periaxin. Again, a very new paper supporting that periaxin levels in the blood can relate to pathology. Neurofilament light levels that we use here is an axonal degeneration biomarker, and again relates to the severity on the improvement of the disease. So definitely you cannot replicate biopsies in patients. Yeah.
Mary, you have a question?
Yeah, I have a follow-up question. Thinking forward to the clinical trial planning, I don't know if you have any kind of thoughts on this, but what would the placebo control arm look like for this?
So we are aiming in a non-placebo control group in this study, and this is something that we have seen in other rare diseases before, that is not unethical to include a placebo group in a rare disease patients group that has to do with viral injection. So we are aiming in a non-placebo group.
So using the natural history data.
Mm-hmm.
Got it.
Yeah.
Thank you.
And CMT1A has a very detailed natural history data.
Yeah, very good. Thanks.
Yeah.
I'm also curious. I'm also curious, do you think that correcting the Schwann cell pathology will be enough to normalize any kind of long-term axonal health issues that might be independent of the Schwann cell dysfunction?
Well, so you can improve myelination, but if the axon is lost at the moment, we know that you cannot get it back. So that's why we're aiming from the earlier intervention possible. I don't know if this answers your question.
Yeah, I guess so then follow-up question, when would you anticipate ideally being able to administer this treatment in patients?
This is not a question for me. This definitely has to do with financials. So we need, just for you to get a very quick impression, in order to apply for the clinical trial stage 1 and 2 that we are aiming for CMT1A, you need to have the product to be injected in humans ready. When we're talking about viral products, this is a very super expensive asset. And if we're aiming for like 20 patients, this has to do with millions. And this is just one thing that is needed starting the trial. And then you have to cover all the monitoring of the patients and many other things that are needed to initiate the clinical trials. So at the moment, the only thing that holds us back is the lack of funding. And we're looking for big sponsors to buy tickets. And the project now is licensed by Armandus Bio, that they are handling all these financial and administratives around the clinical trial initiation and the funding collection.
Okay. We have another question in the chat. They asked, how are you measuring PMP22 expression after treatment? Have you found skin biopsies useful?
We haven't used them. So the skin, you mean if we use them?
Yeah, I think if you have used them is what
We haven't used the skin biopsies in mice or the NHPs. But there's a paper already published from John Svara's group proving that they're useful in patients.
I can ask another one. So you saw that the AAV9 vectors didn't exacerbate any inflammation that you saw within the nerves. And I'm also curious if you've looked into any evidence that correcting the PMP22 expression alters Schwann cell secretion of chemokines and influences that axonal health or the local nerve microenvironment outside of any effect that the AAV9 might have on the inflammation.
So we haven't checked the chemokines or anything about this one cell, but and the first part of the question was
Oh, I'm just wondering if this treatment that you're giving with the vectors has any impact on the nerve microenvironment and inflammation that is seen within CMT1A. And I was just acknowledging that the administration of the vectors themselves didn't cause any inflammation.
so in mice, in the JCI paper and our molecular therapy paper, we know that the nerves of the CMT1A mice, they do have at baseline increased inflammatory CD markers. So there's inflammation due to the pathology. And the AAV9 vector improved this inflammation of the nerves. So it was part of the pathology and it was improved after a treatment. Now, the AAV9 also reaches the liver, and we have analyzed that also in mice in the different doses and different time points, 6 weeks post-injection and 4 months post-injection and 8 months post-injection, all in mice. We have seen that there's a transient inflammatory response. So we know that when we inject the AAV9 and analyze the animal at 6 weeks, there are increased levels of CD markers that they're then normalized back at 4 months. So there's transient inflammatory response, at least in mice, but we have seen no inflammatory indications in the NHPs.
Thank you. Okay, another question in the chat. Do you know how much of the observed functional rescue is driven by reducing newly synthesized PMP22 versus clearing the existing misfolded or aggregated PMP22?
We don't know that.
I am curious about the studies that you saw with the length of how far within the nerve that you are seeing this vector genome. So it's really exciting that you're able to see it in the proximal and the distal ends of the nerve. And I'm curious kind of what the difference in length is between these non-human primates and then humans, and if you anticipate any issues or have any concerns with being able to achieve this correction in the length of the nerve within a human.
Again, I will refer back to the SMA story. Yeah, I mean, we tested that in NHPs. I mean, the perfect model is always a human model, but unfortunately, before going there, we need to do everything and get as many answers we can from models. A monkey, it's like 3 kilo monkeys, to give you an impression. The macacas that we use is a 3 kilo animals. It's bigger than a mouse, but it's definitely not a human.
Would you ever anticipate wanting to move into a like non-human primate model that's a little bigger to try and assess that? I don't even know if that's really possible financially and everything, of course, but
And there are also ethical concerns. So I'm not sure if we're allowed to move in any bigger animals because they have conscience after a while.
I see. See any more questions?
Mary, you had a question?
Yeah, your data on the NFL was really, really nice in the mouse model. Can you comment on the NFL change in the human and with the therapeutic, how long would you expect to have to wait and treat to see some effect on the NFL?
In humans.
Mm-hmm.
So we generally do not believe that there can be readable changes very fast. CMT1A is a slowly progressive disease, and these markers, they're slowly responsive as well. If we were to plan, I think we wouldn't test them earlier than 6 months before. I mean, definitely months, not days and not weeks, and definitely every year.
Got it. Have you done any biomarker work to look for potential other disease biomarkers that might correlate with PMP22 reduction?
Yes, we do have a new project that is done in CMT1A mice, and it has to do with proteomics and limitopics. So we are planning to do a very first discovery in the animal models, and then see and check if what is suggested in these models is also true for humans and back. I mean, as I told you before, a very new suggestion is that periaxin that is found in the blood is a good, let's say, pathology readout.
Cool, thank you.
Thank you.
Just a quick comment for an open question, or more of a comment from Meg. Have you thought about sheep or mini pig models? Have those been used in the context of 1A or with some of these viral vectors to test, like in terms of biodistribution or things like that?
Yes, we have thought about creating a pig CMT1A animal, which is a very bigger animal, and definitely we could test therapies there. But again, everything was stopped when finding out how much this would cost. That was much more expensive than NHP's project. So creating a pig CMT1A animal and testing the therapy, there were a few millions. But that would be ideal if we had all the money of the world.